What's up, friends on the internet and the people in my class? So, first thing we're going to work on today is a pedigree. So, who knows what a pedigree is?
It's just a chart showing how traits are passed on.
Yeah, it's like a family tree with a specific trait to see how it's passed through that family. So, on the board here, I have a chart for albinism. And so the people that are shaded in, they are albinos. Remember, the squares are boys, and the I was going to say Kirby. The circles are girls. So, and when you have a line, these are individuals that, when they connect together like this, these are individuals that are having the offspring below them. So we got 3 different generations here. We've got some different people that are albinos, and then some of them that are not. So what I want you to do from looking at the chart is I want you to try to make a determination. Is albinism a recessive or a dominant trait? I don't want you to look in your book for the answer. I don't want you to figure it out by looking at the chart up here. And then I want you to assign the genotype or possible genotypes that should go with each individual. Okay? So you're going to work through that. Think about, okay, is it recessive? Is it dominant? And then what are the different genotypes that are going to go with each one of these? Okay, so I'm probably just going to use A's, big A's and little A's. So first thing I do is figure out if that's recessive or dominant, and then you can go from there. So I'll give you a hint here. We'll say all of our big A, little A, or little A, little a. So those will be our 3 genotypes, so we all use the same ones. And then you got to figure out which one goes with each. If you can't narrow it down to just one, then put like 2 of them and put a slash in between it. Okay? I'll work on that. So we're going to come back to the pedigree toward the end of class. Once we get through talking about the notes, make sure we understand a few other concepts before we get there. And then we got a really big pedigree that we're going to work on the royal family of Europe. I think it's Queen Elizabeth's family, and there's about 4 different generations. And if you know anything about the royal family from the 17, 18, early 1900s, there's a lot of inbreeding going on. People running around with 6 fingers and hemophilia everywhere. And there's drawbacks to inbreeding, keep it in the family, so to speak. So we'll discuss some of those issues. So if you turn to 287 real quick, on 287 there's a picture. Pretty graphic, I'm going to warn you here. Okay. So we've got a lady fixing to have a baby here, or she's pregnant with one. And on one side, they're doing an amniocentesis, which is where they basically stick a shot in there and take some baby tissue. And on the other one, they're using the, make sure I pronounce this right, chorionic villus sampling. And so they're sampling for cells in the cervical area there. And what they're basically trying to do is to get some DNA from the baby so that they can do a what? What is the picture of? What?
A genetic test.
What's the genetic test?
Karyotype.
Yeah, they're trying to do a karyotype. So what does that karyotype tell us about that baby?
Tell us if there's any genetic mutations.
It doesn't tell us any genetic mutations, but it could tell us what?
Chromosomal.
If there's any chromosomal abnormalities, yes. So you can see there. Now, when we speak of chromosomes, there's 2 different types of chromosomes. What are they? Autosomes and sex chromosomes. So autosomes are numbers 1 through 22, the pairs 1 through 22. And then the sex chromosome is pair 23. So make sure you keep note of that, because you're going to use different methods of determining how traits are passed on depending on which chromosomes we're looking at. Okay. Now, one other thing that I want to remind you of is this weekend, I want you to make a family tree. And you should be able to go from parents to grandparents and probably some aunts and uncles. And I imagine if your grandparents are still around, or maybe even if your parents, I'm sure your parents remember who their grandparents are, that you should be able to go back at least 3 generations, unless there's something strange or different happening there, in which case you can let me know. And then as many of their brothers and sisters as you can put on there, I would like for you to put on there. So you're going to make a pretty big family tree if you can. And you'll just have to ask, like mom or dad or aunt or uncle or grandma or grandpa, and try to find out if these people had any type of genetic disorders that could possibly be passed from one generation to the next. So a genetic disorder, it doesn't necessarily have to be something that killed them, but it could be something, that is significant. This morning, somebody in class said, like, my dad has color blindness. And so we're going to go through in a second and talk about color blindness and why that's important to note. Now, but if you wind up with color blindness, usually that won't kill you unless you got one of those weird stoplights that's sideways and you can't tell which one's red, yellow, or green, and then that might be problematic. But so those things are important, and I think for you guys, it's important to know, like, who in your family is there a family history of heart attacks or stroke or diabetes or what else am I missing? Some other stuff, hemophilia, cystic fibrosis, a number of things, alcoholism, things that we consider that there's a genetic component to, okay? And so I'm just going to kind of check off and see that you've done that. But that's more for you guys as you get older, things that you should be aware of, breast, colon cancer, things like that, that there's a strong genetic component, and things that you should be aware of. Also, things that if your parents haven't considered them, maybe things you should make them aware of. Be like, hey, your grandma and your grandma's grandma or your aunt both had breast cancer, mom. Maybe you should go tell the doctor and get checked out before you get too old, because that kills a lot of people. So those are things to look out for, especially as you guys get older, and if you're knowledgeable about the topics we're discussing and maybe your parents aren't as knowledgeable, that's something you can discuss with them, okay? So anyway, let's go through here. Linked genes. Let's talk about genes and what that means to be linked. So if you turn to page 304, 303, 304, there's a map on here, a gene map for Drosophila. Drosophila are fruit flies. Fruit flies are studied a lot in genetics for what purpose? You might know?
Lots of variation.
Okay, so there's lots of mutations. There's only 4 chromosomal pairs. So there's not a lot of DNA. I mean, it's pretty easy to study those 4 chromosomal pairs. There's a lot of mutations. Nobody really cares that much if you raise some fruit flies, kill some fruit flies. It's not like, cats or bunnies or monkeys or something like that. So if you want to raise a bunch of fruit flies, have their babies, see what their offspring are, have their babies, and then you kill the ones previous, then it's usually not that big a deal. So, and they have generations really quickly. So in a few weeks, you can have like a new generation. Maybe it's 4 weeks, 3 or 4 weeks. You can have a whole new generation. So you can look at the offspring. Now, the lab that we're supposed to start tomorrow, we'll see, I'm sure we'll get a little bit started on it, is supposed to look at the genetics of fruit flies. And so I'd originally ordered this back in the day when I started doing this lab. And then I realized how much of a pain in the butt it was going to be to try to mate fruit flies. And I decided to use some experimental data that came from one of the lab websites that I use. So we're not going to be raising fruit flies. I'll be honest with y'all. I had fruit flies. I would see them around here for like probably 7 years after that initial group. I don't know what they were eating, how they were breeding. But those jokers, some of them got out that first go around, and I never did get them sorted out. So anyway, watch out for those fruit flies. All right. Linked genes. Walker, read that for us.
These are usually inherited as a linked unit because they are found on the same chromosome. This term usually is associated with genes on autosomes 1 through 22.
Okay. Now, it's going to depend on what organism, but for us, yeah, it would be on 1 through 22. That term, linkage, let's skip down to the next one. Alfred Sturtevant was this like research assistant to some guy named Morgan, and he's the one that figured this out. It's on page 303 in your book there, about how to construct a gene map. A gene map, all it is it's showing you relatively where different genes are on a chromosome. So there's this term here called linked genes. What does that mean to be linked together? connected some way. So now this isn't exactly one, but I'll give you this example just so y'all can wrap your heads around this. A lot of times we think of people with like blonde hair and blue eyes inheriting those things together, right? So those are two things you hear matched up. So let's say, for instance, there's hypotheticals here. We're going to say that blonde hair, blue eyes is linked together. Where would you expect to find them on a chromosome?
Closer to the centromere?
Closer to the centromere and relative to one another. Close together. So you would expect them to be close together and probably be close to the centromere. And so in that way, those two traits would be inherited together. And they would be less likely to be affected by crossing over. Good. So when you talk about linked genes, they're usually closer together, a lot of times closer to the centromere. The main thing is they're less likely to be affected by crossing over. Okay. So in the next part here, he used crossing over rates to determine loci on chromosomes. Remember, loci or locus is just short for location. The finished product is a linkage map, so showing where things are, just like the one on the fruit flies. I'll pull it back up here in a second. The smaller the rate of crossing over, the closer they are together on the same chromosome. So if you're looking at two traits and crossing over doesn't seem to affect them that often, then usually they're closer together on a chromosome. Okay. The higher the rates of crossing over, the farther apart they are, the more likely crossing over is to affect them. Okay. And so there's kind of this arbitrary unit. I'm not really worried about that. But the main thing is that's how they came up with this gene map here, showing you where some genes are relative to others. So if we look here on this chart, it's important to note here, wild type phenotypes. What does that mean, wild types?
Occurs naturally.
Yes, so those are the natural phenotypes. Mutant phenotypes are mutations, okay, that don't occur naturally. So fruit flies have been bred in captivity, and people keep breeding them, and some weird stuff happens. They just have a lot of weird mutations. So let's take an example. The cinnabar eyes and the black body. What would you say its likelihood to be linked together is? Cinnabar eyes and black body. You think those genes are normally linked together? Yes or no?
Yeah.
For the mutant type. Yes. Why?
They're close.
They're close together on the same chromosome. What about short antennae and brown eyes? No, they're far apart, and they're much more likely to be affected by crossing over. So basically what Sturdivant had done, so you don't have to read that whole spiel, although it's pretty interesting, is they went through and they'd bred all these generations of fruit flies and looked at all these characteristics. And so he was looking, y'all know how on the lab we did with Sordaria last week, y'all had to figure out the map units and the crossing over rates. So he went back and looked at that and compared those between the different genes and saw like how different they were or how similar they were. And so the less likely they were to be affected by crossing over, the closer they were together. The more likely, the farther apart they were. Okay? So if you hear anything about gene maps, that's what it's talking about. Where on the chromosome is it located relative to other genes? Next thing, sex-linked genes. Turn over to page 304. No, hold on, that's not right. 296. What is a sex-linked gene? All right, a gene found on the X or Y chromosome. Good. Typically, most sex-linked disorders are found on what? The X. I'll be honest with you, on 296 here, if you look in the X and Y, what do you notice about the Y? It's what? Small, yeah. It looks like, you got the huge X over there, and then that Y looks like a dingleberry that fell off of it. So there's not a lot to it. And in fact, there's not a lot of genes that go with it. So let's go through and we'll look at this here, and then we're going to look at some disorders and how these sex-linked disorders actually work. Remember, boys, we get an X and a Y, and then girls, you have two X's, okay? So I know that makes us more awesome because we have more different genes, but it's actually problematic for guys because they're the ones usually afflicted with sex-linked disorders. The fortunate thing for ladies is you have two X's, so if you get a bad one, what happens? The good one usually backs it up. Now, you could get a sex-linked dominant disorder, which means it's going to show up whether you have two or not. If you have a good one, a good normal gene. But most of these sex-linked disorders that we're going to look at are sex-linked recessive, meaning it's found on the X chromosome and it's going to be recessive if you have a good normal X. Unfortunately for boys, we don't have another X to cover up that bad X if there's one there. So we usually wind up getting that disorder. So that's why men are much more afflicted with those sex-linked disorders, okay? A couple things to note there. Part B, sex chromosomes undergo very little crossing over during prophase. Sex of the organism will be determined at conception. Now, this is true for us and most mammals and some other stuff, but a lot of reptiles and amphibian and fish, a lot of them, their sex is determined by, it's not on X and Y, what else could it be determined by? A lot of organisms are temperature dependent. So like sea turtles, when they lay their eggs, they lay them in a little curvy shaped hole that they dig out with their flippers. And the ones that are toward the top, the ones that get hotter, those are the females. So, I mean, that could be a good thing where if you're global warming, things get warmer, sea turtles are endangered, it gets hotter, so there's more girls, and then you have more girls, you don't need as many guys, you can have more babies. Now, from what I looked up yesterday, not all organisms that are temperature dependent are the girls are warmer. Sometimes it's the opposite. The guys are the warmer ones. So it really is dependent upon species. So that's something you'd have to look up for different things. So some things it may be less beneficial to as like climate change occurs and affects us. All right, let's see here. But we are XY determinant. So if you get an XY, you are biologically a male. And if you're XX, you're biologically female. Now, I'm not going to get any political discussions here on this, on other topics related to this, but that is things, those are items that we know for sure. Now, sometimes you may have an XY and you may have someone who turns out to be a hermaphrodite, or some people would say intersex. Have y'all heard these terms? So what happens in that scenario? They have features of both. Let's skip down and look at part D here. Everyone starts out female. Where is that? Hold on a second. There we go. Everyone starts out female. This is why we have nipples. I did not write these notes, but anyway. The blank slate person is going to be a female starting out. And then about 2 months of age, the Y's SYR gene, SYR stands for sex determining region of the Y, starts production of testosterone and the male sex organs form. After development is complete, testosterone is turned off until puberty, and when you develop secondary sexual characteristics such as facial hair and all the other weird stuff. So you can imagine a scenario where maybe that gene didn't turn on properly or all the way, or maybe late, and female organs were already in development. So it says in your book on 296 on the bottom right, in the absence of the SRY gene, the gonads develop into ovaries. So even if you have an XY chromosome, then you can develop ovaries in the absence of that gene. So if there's a mutation in it or it doesn't switch on, which I would think would probably happen from a mutation, but anyway, so it's not going to code for those proteins. It's not going to, testes aren't going to develop in those male characteristics that we see, okay? And then you wind up with someone who's a little bit on the in-between in development. Historically, for the past, 60, 70 years that happened, what happened when you came out at birth and you had both organs partially or somewhat developed? Usually your parents, picked one or the other sex for you, and usually a lot of times they had surgery and like it kind of picked for you to be male or picked for you to be female, and that's what you got stuck with the rest of your life. So there is some iffiness there in terms of like male and female biologically because of development and something that may go awry during those early stages of development. But I would say that's limited to a very small group of people. I don't know the percentage, but I imagine it's very, very small. Okay, so where do we go now? After patterns of inheritance and some sex-linked disorders. So before we get into that, let's look on page 297. So I'm going to read a little something for you guys just to kind of give you an idea of a little more information about the X and Y chromosome. Okay? So, at the top of 297, researchers have sequenced the human Y chromosome and identified 78 genes that code for about 25 different proteins. Some are duplicates. So of all the genes we have on our X chromosome, fellows, they're only going to code for about 25 different proteins, which is a very small amount. If you skip down on the X chromosome, okay, we have an X too, so we can partake in this. Chromosomes contain approximately 1,100 genes. So 1,100, 78, y'all can see there's a pretty big difference there in the genes that are located on those. Okay? Interesting. On 297, let's talk about some sex-linked disorders. So, some you may be familiar with here. For example, color blindness. Color blindness is where your body doesn't produce, you're not able to produce one or more of the three cones that we have. Red, green is the most common, and like I said, if you're at the red light, if you have a red, yellow, green, that's okay because you can see 1, 2, 3. If they turn that joker sideways, it might be a little more confusing. I mean, usually they're red left to right, but I could see how just like looking off the cuff or driving down the road and it changes, like it might freak you out. So one of my students in first period, whose initials, I'm not going to say, never mind. She said, hey, my dad's colorblind. And then I said that about the red lights. And she's like, yeah, he does hate those red lights. And I was like, yeah, I probably would too. So that's something to watch out for. So you guys need to make a little Punnett square on your Punnett square practice sheet. And let's go through and do a little Punnett square. I want for you to cross, let's say your father, okay, so in your Punnett square, the dad has color blindness. Now, this is a gross oversimplification. There are many different types of color blindness, but we're gonna make it simple here. Okay, so we're gonna just say one thing: color blindness. And to be honest, most people they're not missing like all three, which would mean you'd like see everything in grayscale, which would be interesting maybe for a day. But after that, the whole black and white scene, it's like you're in a movie from the 50s. But anyway, so we're just gonna treat it like one thing is happening. So we've got a dad who is colorblind and a mother who is a carrier for colorblindness, but she is not colorblind, okay? Now this is I'm going to go ahead and tell you, it's a sex-linked recessive trait. So complete that Punnett square. Okay, when you're doing a Punnett square for sex-linked disorder, you're not going to use just one letter, okay? So here's how you want to set this up. We got dad. We got mom. Okay? So we said it's sex-linked recessive. The dad has color blindness. Color blindness is going to be found on the X chromosome, so we're going to put a little C there. Okay? The mom is a carrier, so one of her X's, she has to have a C on. Okay? Now, you could put an N right here for normal cones or whatever, I don't care, but I'm just going to put a C for color blindness. We'll assume this one is normal. Okay? So, and then you're going to do that cross. How many of y'all set this up like this? Nobody. Okay, that's okay. We'll go ahead and fill this in, and then let's draw some conclusions. So, since the C is recessive, I'm going to put it second always here. Unless you got two of them. All right. When you fill that out, how many of, let's say that these are your offspring you're looking at, Ashlyn. What's the chance of you having a boy that is colorblind? What? 50%. So you're going to look at this a little bit differently than we normally do. We're just going to look at it from like boys and girls because they're going to be different. What's the chance that a girl you have would be colorblind, Sarah? 50% too. So this one's colorblind, this one's colorblind, okay? Not colorblind, carrier. So in actuality, I think from our person this morning, I don't think her mother was a carrier because none of the boys wound up having color blindness, and you would expect out of 2 or 3, you know, a 50% chance it would show up. And so probably her mother's not a carrier. But the problem for her would be what? If you're a girl and your dad is colorblind, what's going to happen to you for sure? You're going to be a carrier for color blindness. So that means you're going to be a carrier, and that for your offspring, there's going to be a what % chance that your boys are colorblind? 50%. I've only ever met one colorblind girl. You 3 people out there watching, if you're one of them in the comments, y'all can write it. And it was a funny story. It was an older lady who worked at a clothing store, and I was like, that's a really bad idea, I think. But anyway, don't discriminate. So, but anyways, that's how you're going to do a sex-linked disorder chart. Now, let's go back and finish the notes, and we're gonna practice a big pedigree chart here in a second. So the last thing that we've got on the notes, is hemophilia here. Hemophilia is like a free bleeding disease. Basically, you're not making enough platelets, and you can't stop bleeding. You can bleed internally, you can bleed externally, you can bleed out that way much more easy than a normal person. Just a normal bruise could kill you, or something that would cause a normal bruise for most people could kill you, so it's a big problem. And this was prevalent in the royal family of Europe. Okay, like I said, a lot of inbreeding is passed throughout, so that's what you're going to be working on today. Now, nowadays we have a shot called AHF, anti-hemolytic factor, that you can give people that are experiencing this. So we do have some treatment. So, any questions there? All right, so here's what we're going to do. We're going to finish up the pedigree that we started at the beginning of class, and then we're going to work on the royal pedigree. And this is all over the internet, but I'm going to put a picture up here so you guys can look at it for the 3 of you not in my class at school if you want to work on it, because it's really good practice for how to work these pedigrees. Okay, so albinism. Were y'all able to narrow down most of these individuals and what their traits were? Yeah, I don't hear a lot of confidence here. Which ones do we? Okay, so out of our three genotypes, which one are albinos? The little A's. Okay, so this is a recessive disorder. How did you know it was a recessive disorder?
Because it isn't.
I'm going to put NC right here just for like normal color, whatever your normal color you would normally inherit, whether it's light or dark or whatever, you have some pigmentation. And so albinism is where you don't inherit those, any of that ability to make pigmentation, the melanocytes. So, what did you say? How'd you know it was recessive?
It wasn't dominant.
You just knew? Anybody? How would you tell by looking at the pedigree that albinism wasn't dominant?
Because not every kid.
If it's both little A's, then that means the parents have to be big A, little A, and that's the only way you could come out with it.
Yeah, you couldn't have a kid that was albino and have 2 parents that weren't albino, right? Unless it's recessive. If it's a dominant trait, every time an individual has that, at least one of their parents has to have it. So that's the key distinction on a pedigree. When you're looking like this individual's has albinism, then they've got to have a parent. Well, okay, well maybe that's the case, but if you look at this parent, then neither one of them do. So they're getting these genes from them, and it has to be a recessive trait. So, albinism. So the easy ones to do are what? First ones you fill out?
The colored ones.
Yeah, they're all going to be little A's. Okay? We know that much. We're good there. All right. Now, let's say, what about all of these other offspring down here?
Big A, little A.
You can for sure narrow it down to big A, little A. Why?
Because they're not albino, but they have.
What about the mom here?
Big A, little A.
She's got to be big A, little a, because if these individuals are getting a little a from each parent, you can only get one little a over here from this parent. You've got to get another little a from this parent. And we know that they're not albinos, so that they've got to have a big A, but they've also got to have a little a from this parent. So all of these individuals are big A, little a. Okay? Does that make sense? All right, now let's go through some others. This individual's parents up here, what are they going to be?
Bigger, little
Okay, they're also going to have to be big A, little a, because we know they're not albinos. Okay, they don't have albinism, and they have to be a carrier in order for this offspring to have two little a's. So we can narrow it down. What about this individual over here? What are they going to be?
Big A, big A.
Big A?
You don't know.
You don't know. It could be big A, it could be big A, little a. We know they're not little a, right? But they could have inherited either or. So we don't know. What about this individual's parents up here? We know that one of them has to be a carrier. That's really all we know. We don't know which one. One could be a carrier, one could be big A, both of them could be carriers. We don't know. So we'll just put both, and the same thing over here for this other daughter. We don't know which one she's going to inherit from the parents. Okay? So when you don't know, you need to put, there's two possibilities. If you do know for sure, then you need to narrow it down to what that possibility is. Okay? Does that make sense? So today, for those of you out here, this is what we're working on. And this chart's on the internet. It's Pedigree chart, Hemophilia in the royal family. So it starts out with Queen Victoria. Maybe I said Elizabeth earlier, sorry. And it works its way down here. You may know this, Alexei Tsar of Russia. Y'all ever heard of Rasputin?
Yes.
And so Alexei had hemophilia, and his mama hired this dude, Rasputin, to kind of take care of him. He's kind of a witch doctor. Sorry if some of your relatives are out there. Anyway, there was a coup. They shot them all and killed them all. And Anastasia was one of his sisters. They thought she might have got away because they couldn't find a body. There's this whole big hullabaloo about it. But anyway, so this is that family. But there's people from all over Europe in this. So what we're going to do today, you're going to know the people that have hemophilia. They're shaded in. But all of these other individuals here, you've got to figure out what they are. Okay? And then where these traits got passed down. Okay? So like I said, there's a lot of inbreeding going on, but we're going to work our way through this. So there we go.